Welcome to Te Whare Wānaka, Aoraki, to Lincoln University. It gives me great pleasure to see such a diverse array of individuals in the audience, but also such a large audience, especially for this excellent series. And I think Katie started early because she knows Dee can talk.
Hope you brought your dinner.
So the Excellence Series celebrates outstanding scholarship and research that's helping shape a more sustainable and resilient future for Aotearoa New Zealand. It brings together leading researchers whose work challenges us to think differently, sparks important conversations, and connects science and innovation with the real world. Today, we're delighted to welcome Professor Hong Dee. Professor Dee is internationally recognized for his contributions to soil and environmental research, particularly in the areas of greenhouse gas mitigation, nutrient management, and water quality in agricultural systems. As head of the Center for Soil and Environmental Research, Professor Dee has led major national and international research programs funded by organizations including MPI, the Global Research Alliance, and industry partners. His work has significantly advanced our understanding of nitrogen cycling in agroecosystems and the role of soil microbial communities in nitrate leaching and nitrous oxide emissions. Importantly, his research has gone beyond theory and into practical application, developing technologies and mitigation tools that can help reduce agricultural greenhouse gas emissions and nutrient losses to waterways while supporting productive farm systems. These innovations are helping address some of the most pressing environmental challenges, both to New Zealand, but globally in agriculture today. Professor Dee's contributions have been recognized through numerous honors, including being appointed an officer of the New Zealand Order of Merit for services to agricultural research. He's also a fellow of the Royal Society of New Zealand, the New Zealand Institute of Agricultural and Horticultural Science, the New Zealand Society of Soil Science. Alongside Professor Keith Cameron, he was awarded the Pickering Medal for the development of ClearTech, a reflection of the real-world impact his research and innovation has had. In today's seminar, Professor Dee will share insights into the technologies his team has developed to mitigate greenhouse gas emissions and reduce nitrate and phosphate contamination of waterways. He will discuss the science underpinning these technologies, how they were developed, and the potential they hold for improving environmental sustainability of grazed pasture systems. This is an important and timely conversation as we continue to explore how research, innovation, and practical solutions can help support both the future of farming and environmental stewardship in Aotearoa, New Zealand. Please join me in giving a very warm welcome to Professor Hong Dee.
Thank you, Chad, for that introduction, and a very warm welcome to all of you who are here and those online as well. I hope you can all hear me okay. I do have a very loud voice. I was trying to become an opera singer when I was younger. So today, I will give you a very high-level overview of some of the work we've done over the past decades, in fact, in trying to mitigate greenhouse gas emissions and nutrient losses to waterways from the agricultural system, or grazed pasture system. First, I would like to acknowledge my friend, my colleague, Professor Keith Cameron, who is here today, for his huge contribution to this work. This is really a joint team effort. Keith is here today to answer any current questions. I would also like to acknowledge the team in the Center for Soil and Environmental Research, and particularly Dr. Andre Padolian, who is a senior research officer in the center, for their contribution to the work. And also acknowledging the funding from government, from industry partners, and from Lincoln University. The global temperature is definitely rising. So this graph shows you the temperature rises above the pre-industrial average. And you can see in 2024, we in fact exceeded the 1.5 degrees threshold. And the consequences as a result of these temperature increases are felt all over the world, including New Zealand. Based on the UNEP projections, and you can see if the current policies continue around the world, we are almost certain to exceed the 1.5 degree increase above the pre-industrial levels. We probably will also surpass the 2 degree threshold. So really there's pressure for governments around the world to basically really what they promised at the Paris Agreement and maybe doing some extra stuff to reduce the greenhouse gas emissions so that we can keep the temperature rises to certainly below 2 degrees and preferably below 1.5 degrees. So that is the challenge. Current policies, we are certainly exceeded the 1.5 degrees. Looking at the New Zealand greenhouse gas emissions profile, I'm sure many of you are aware, we are very unique among developed countries in that about 50% of our greenhouse gases come from the agricultural sector, particularly methane from the rumen livestock, and then also nitrous oxide being the second largest greenhouse gas. So New Zealand has made commitments under the Paris Accord, and increasingly, the greenhouse gas emissions from the agricultural sector is becoming a trade issue, market access. Big companies like Nestle, Danone, they are asking the suppliers to reduce their greenhouse gas footprint. The other environmental issue that is related to the agricultural sector is the water contamination by nutrients, such as nitrate leaching, phosphate leaching, E. coli leaching, contributing to the surface water quality deterioration and drinking water contamination as well. So we've been doing research for a number of years, firstly trying to identify, to quantify where the contaminants come from. So we've done many experiments trying to source, to characterize the source of nitrate and nitrous oxide. I show you a couple of diagrams here. You can see the nitrous oxide, for example, is a lot of it is actually coming from the urine patches. So if you go from a sheep to maybe a beef cattle to a dairy cattle, as the nitrogen loading rate under the urine patch increases, the nitrous oxide emissions also increase. It's a very similar picture for nitrate leaching as well. Nitrate leaching from a dairy cow urine patch at 1,000 kilograms per hectare is much higher than urea even applied at 400 kilograms per hectare. So the dominant source for both nitrate leaching and nitrous oxide in our grazed pasture system is the animal urine spots, whereas the animals concentrate the nitrogen from where they grazed the pasture and deposited them in a single deposition. That's where the problem is. In terms of phosphate, Professor Mr. McDowell and others have done some research on the Lincoln University dairy farm, quantifying the phosphate leaching losses from the areas under the effluent applied area on the dairy farm. And so over a 14-year period, you can see where the effluent is applied, particularly to a free draining soil, such as an ear soil, which many of you are probably familiar. There's a significantly higher phosphate leaching loss from that area than from the rest of the field. So farm dairy effluent is a major contributor to phosphate leaching and runoff as well to waterways. So how do we mitigate these losses? That's the key question. Our cows are very well behaved, but they're not so well behaved. You can train them to urinate in a particular spot, for example. Maybe achievable in the future. Certainly, at the moment, they seem to urinate and defecate wherever they like and whenever they like. So Keith and I are soil scientists. We try to find the solutions in the soil. So this is a simplified nitrogen cycle in the soil, in the grazed pasture system. When the urine and the urea fertilizer, for example, is dropped onto the soil, it first releases ammonium. Ammonium, you may or may not know, that undergoes some transformations. It goes through a nitrification process, usually through some multiple steps, producing nitrate. A nitrate, being a negatively charged ion, is not retained by the soil particles, stays in the water, so whenever there's drainage, nitrate leaching occurs. And nitrate can also be denitrified in the soil, producing nitrous oxide as an intermediate product. And the nitrification process also produces nitrous oxide. So nitrification, denitrification, producing nitrous oxide, and nitrification producing nitrate, which contribute to leaching. So we had a hypothesis, and that is, what if we treat the soil with a nitrification inhibitor? So a nitrification inhibitor does is it slows down the first step of the nitrification process. Either slows it down or stops it maybe temporarily, keeping the ammonia, nitrogen in the ammonium form for longer, so the plants can utilize it. So by doing so, you are slowing down the nitrification process, and therefore you reduce the nitrous oxide from the nitrification process. You're also reducing nitrate concentration in the soil, and thereby reducing nitrate leaching as well. And because of the lower nitrate concentration in the soil, it also reduces the nitrous oxide from the denitrification process. So the key is to basically stop or slow down the first step of the ammonia oxidation. But the microbes that are involved in this process are very complicated. Okay, if you read the scientific books published 20 years ago, they would say the microbes, the ammonia oxidizers, they are ammonia oxidizing bacteria, AOB. So that's the dominant microbe that does their job. And then in about 2005, 2006, there was a paper published in Nature. It's actually a seminal paper showing that in our soil systems, there is another very important group of microbes called archaea. They are ammonia oxidizing archaea. They also carry the same functional genes, ammonia monooxygenase gene. So they can also perform the ammonia oxidation. And then about in 2005, 2015, 16, another paper appeared in Nature saying, oh, in addition to the AOB and the AOA, there's a third group called a Commonwealth, complete ammonia oxidizers. And they can perform this nitrification process by themselves without going through these different steps involving different microbial communities. So the question then arises, which microbial community is responsible for the ammonia oxidation? How abundant they are in the soils? And can we actually inhibit these microbes to reduce nitrous oxide and nitrate leaching? So to answer this question, then we started a series of investigations. We first sampled the soil, dairy pasture soils in particular, from across the country, from Northland, the Waikato, to Southland, Canterbury, Rotorua Lakes, Canterbury, West Coast, and Southland, to see what microbes actually there, what ammonia oxidizers are there in the different soils? Are there differences between the different regions? So what you can see here, in some soils, such as in the Northland soil, the bacterial archaea population abundance are relatively similar. But in some other soils, like in the central North Island, Waikato and Rotorua, there were more archaea than bacteria. But in Southland, the soil we collected, there's a lot more bacteria than archaea. Because this was, very new, we had no idea why there are such wide variations. So knowing that they are both there, so which microbe is actually responsible for the ammonia oxidation then? So we started a series of investigations under controlled environments to look at how the microbes respond to additions of nitrogen, to the variations in environmental conditions, temperature, moisture, etc., to understand the microbial community behavior. So here is an example showing the bacteria AOB population abundance responses to the addition of animal urine. And you can see when you apply the urine, the population abundance of this bacteria community really took off, just exploded. So the abundance increased very dramatically because the urine supplies ammonium, and ammonium is used as an energy source for the bacteria to grow. As a result of this rapid increase in population, so the bacteria in turn can also rapidly transform the ammonium into nitrate. So the nitrate ammonium disappears quickly and nitrate increases. And then we thought, what if we put a nitrification inhibitor in there? So you can see the urine plus a DCD, which was a nitrification inhibitor. You can see the abundance did not increase at all, even though ammonium is supplied, because the inhibitor has essentially blocked the active site of the enzyme responsible for the ammonia oxidation. They are not able to use ammonium as an energy source. So they were not killed. They just stayed dormant in there. So because the population remained low, that means the ammonium oxidation slowed down because the smaller population. We also looked into the archaea population, the AOA abundance. Very interesting. There's a lot of the archaea in the soil, very high numbers, but they did not seem to care whether you apply the urine or urine plus a nitrification inhibitor. So that's really interesting. They are there, but they're not doing much, even though we applied a lot of the nitrogen in the urine form. So that is the abundance. And then we looked into their activity by quantifying their RNA copy numbers. So how their genes are expressed. And you can see if you apply to the animal urine, the AOB abundance increased and their activity also increased, which means they are really busy converting the ammonium into nitrate. And if you put an inhibitor in there, the activity is also inhibited. And the archaea show the same picture. They did not grow and they were not active, not very active at all. And then we did further investigations. What we found was the archaea, in fact, does not like the high nitrogen environment and a urine patch. They prefer a more low nitrogen environment. So here, if you look at this diagram, the archaea gradually, slowly sort of grew when no nitrogen was applied. Where you apply the nitrogen in the urine or urine plus inhibitor, they didn't change much. And another study showed the same picture. In the control, there was an increase in archaea. In fact, if you apply so much nitrogen, it actually inhibited their abundance. So they do not like the high nitrogen environment. So we subsequently conducted a number of studies where we realized archaea doesn't like high nitrogen environment. They like the low nitrogen environment and also acidic conditions. Bacteria, AOB, they love the high nitrogen environment. So you put the urine in there, they just take off. So if we look at the relationship between nitrate, the nitrification rate, the concentration of nitrate produced versus the AOB population, you can see there's a reasonable relationship. As the population abundance increased, the nitrification rate increased. But the Archaea population, we could not find any relationship at all. So that really tells us for the grazed pasture soils and the urine patch, bacteria should be the target to inhibit. But what about common moles? So we just when we realized, we thought we sort of sorted the microbial communities, and then along came the common moles. So then we did the same thing. We sampled the soil from across the country, quantified the common moles abundance. So you can see in some soils, the common moles abundance was lower than bacteria and archaea. But in some other soils, in the Rotorua lakes on west coast, their abundance is much higher than the bacteria and archaea. Again, because this is all new, we have no idea what is actually affecting their growth and their abundance. So then we took soil samples from different land use types. What we found was that wherever there is irrigation, there's more moisture or rainfall, the common moles abundance seems to be higher. So here, for example, sheep and beef with irrigation. Without irrigation, you can see the common moles abundance is higher in the irrigated system. A very similar picture in the dairy system as well. With irrigation, higher numbers, and without irrigation, lower numbers. So it seems that the common moles communities like the moist environment. And then we also did some incubation studies looking at the common moles sources to the addition of different nitrogen sources. So here is the RA copy numbers. We applied farm dairy effluent, animal urine, urine plus an inhibitor, urea plus an inhibitor. So you can see there is certainly a response in the activity as you apply the nitrogen. But the highest activity was not on the high nitrogen urine spot, but in the urea at 50 kilograms per hectare. So common moles seem to be somewhere in between the bacteria and archaea in terms of the nitrogen environment that they are most active. But importantly, it can be inhibited both under the urine patch and under the urea treated soils. So if they were active, they were performing the ammonia oxidation, it can be inhibited. So bacteria can be inhibited, common moles can, archaea, we can forget about them under the urine patches. So can they reduce nitrous oxide emissions by inhibition? So here are some, I can show you these graphs for until tomorrow. I'll give you a couple examples, one from the Waikato, one from Canterbury, showing you the nitrous oxide emissions envelope after the urea application. You can see there's quite a high emissions envelope if you just apply the urine. But if you apply the urine plus an inhibitor, because of the inhibition of both the bacteria and the common moles, the nitrous oxide emissions were reduced by over 70%. So the hypothesis of reducing nitrous oxide by inhibiting the microbial ammonia oxidizers is basically approved, verified. And then we conducted a range of experiments using soils from different regions under different environmental conditions, different water input, for example. And these gave us an average reduction of 64% in nitrous oxide emissions. So what about nitrate leaching? So here are the nitrate concentrations in the draining water leaching through some undisturbed large soil columns. They are usually 0.5 meter in diameter and 70 centimeters deep. So if you just apply the urea alone, you can see there's a lot of nitrate emerging underneath the lacinities. You put an inhibitor in there, and the concentration is reduced. Similarly, for another soil in Canterbury. And so reducing nitrate leaching by over 60%. We did the same thing using soils from different regions. So we actually went to those regions, collected the last images and bring them back to Lincoln University. We do these measurements under different rainfall conditions, technologies. The approach is very effective in reducing nitrate leaching as well. You may or may not know when nitrate is leached, it takes cations with it. So they are leached in ion pairs. So the nitrate leaching, they usually takes calcium, magnesium, and potassium as companions. The nitrate is not lost on its own. So there's a very good relationship between the nitrogen concentration and this cation concentration. So because we are reduce the nitrate leaching, we also reduce these cation leaching losses as well, because these are valuable nutrients. So by reducing nitrate leaching, we are also saving the farmer some money from purchasing these cations. But there is more. Because we are saving the nitrogen in the soil and saving the cations and some other mechanisms as well, we expect to have an increased pasture production. Okay, so on average, you can probably expect a 10% increase. And Dr. Jim Moyer, who used to work with us in the team, and he's done some measurements even higher than the 10% increases as well. So you get an economic return by implementing some environmental technologies. You get the environmental benefit. And then over the last few years, we have been working with our collaborators in the University of Auckland, Otago, and Ag Research in trying to discover a new generation of nitrification inhibitors. So we screened more than 25,000 compounds, first using in silico screening by using a modeled structure of the enzyme, and to see which compound actually can fit nicely into the active site to block it. From the 25,000, we selected 5,000 to conduct we call a phenotypic screening in the laboratory using live ammonia oxidant bacteria culture to see which compound can inhibit these microbes in the culture. And then from the 5,000, we selected 200 for testing in the soil. We have to work in the soil because that's where it's going to be applied. So from the 200, we are now down to the number one. We have one compound which is more effective than any nitrification inhibitor that exists on this planet. Miles better, and are applied at a fraction of the application rate. So we are just in the final stages of basically getting all the information ready for the release onto the market. I'm not going to go into the details today. Perhaps we can set up another presentation where I can give you the entire talk about the new nitrification inhibitors. Okay, but watch the space. It should come out in the next 2 years or so. Because I have another exciting technology to talk about, and that is the reduction of methane emissions from effluent ponds and phosphate leaching and E. coli leaching when the effluent is applied onto the farm. I'm sure many of you know that all the dairy farms have a huge effluent pond, usually probably at least a million liters, some has 3 million, 5 million liters. You must have enough capacity to store the effluent so that if it keep on raining every day, you cannot irrigate it, you're able to hold it until the weather has improved, the soil is not flooded, so it doesn't cause any leaching losses. So the problem is that during the storage of the effluent in this large pond, methane comes out because there are organic matter in there, and under anaerobic conditions produces methane. So that's the second largest source of methane after the enteric fermentation for dairy farms. And the other thing is that when you then apply the effluent on the farm, you've seen earlier, it can contribute to phosphate leaching, but also E. coli leaching. Keith and I have spent our entire career working on this smelly stuff. Effluent. We love stuff. Yeah. So essentially, the dairy pond is like we call it a large bioreactor. Every day, the farmer, after milking, cleans the yard, cleans the parlor. All that dirty water is washed into the pond and carry with it the organic material, which usually sort of is settled down at the lower part of the pond. And that's where a lot of the methanogens, the methane-producing microbes, they usually are archaea. They love the organic carbon. So we are feeding the methane-producing microbes every day with fresh carbon, and they're producing the methane constantly. If you observe the pond long enough, you'll see some bubbles bubble up. So what we've done is treating the effluent pond with two chemical compounds, iron sulfate and sulfuric acid. There's several mechanisms involved. It can suppress methane production, but I'll list three here. The first, probably a very important one, is these compounds raise the redox potential of the effluent. So if you do not add these, the pond is very anaerobic. It usually goes down to -200, -300 we measured. So under these conditions, the methane producing microbes, the methanogens, they love it. They are very active producing methane. So once we raise the redox potential to about 200 or so, they are dead. It's because under aerobic conditions, they cannot survive. Okay, so they are not able to produce the methane under those conditions. So that's the first mechanism. The second one is that we're adding sulfate. So that stimulates another group of microbes called sulfate-reducing bacteria. And you do not have to look at this equation very carefully. What I can say is that these sulfate-reducing bacteria, they can outcompete the methanogens because they can derive more energy by consuming the same substrates. So essentially, the methanogens are deprived of the substrate. And the third one is that even under anaerobic conditions, the sulfate also enhances some microbes in the pond that can even oxidize methane under anaerobic conditions. So there are at least three mechanisms that make sure not much methane will come out. Does it work? We started in the laboratory using these large columns. So we treat some of them with the iron sulfate and sulfuric acid, and leave others untreated. So you can see if you do not treat it, there's a lot of methane coming out. Okay, you treat it, flat as a pancake. So more than 95% reduction in the laboratory. And we've done a series of experiments trying to establish some relationships. And you can see there's a very good correlation with the dosage of the iron sulfate and the methane produced. The more you add, the less is coming out. So obviously we need to cut it off somewhere for economic reasons. And then we moved from the laboratory to the field using this 100,000 liter effluent tanks. So we treat one of them and leave the other one untreated. We were nervous when we conducted this trial because sometimes you take the laboratory technology into the field and may or may not work. 99.9% reduction in the methane emissions. I'm really sorry, there's still 0.1% left. We've been working on it. There's one experiment we did actually 100% reduction. Yes. And then we thought, well, that's not large enough. Let's measure it under a real pond. So this is a Lincoln University dairy pond. We measure the emissions before the treatment and then after the treatment using these floating chambers, using a laser machine to measure it. And you can see there's a 96% reduction, and a real dairy is basically showing it works. It's transferable from the laboratory to the field. And then MPI funded us a program to basically to assess whether it works in other parts of the country and other dairy systems. So we established 10, we selected 10 dairy farms across the country and installed these large columns. Okay, so each one of them has got 8 effluent columns. They are randomly allocated with treated and untreated to measure the methane reductions. So we can analyze them statistically. And we did the measurements in the different regions and the different seasons. So the average reductions are somewhere around 96 or 97% across the country, to showing that it works in the different environments, in the different dairy systems. You do not need much infrastructure on the farm because the ingredient is carried on the truck. So the truck just comes into the farm and just to deliver the stuff by sucking up some of the effluent through the big pump and inject the sulfuric acid and the PFS through this injection pores, and then returning the effluent onto the pond, into the pond. So and you do not have to treat every liter. You probably need to treat maybe a third, maximum 40%, and because you have a mixing on the farm. So essentially, when you're treating it, the ingredients are mixed with the effluent. It takes about, depending on the size of the pond, how full it is, probably a couple of hours. So you can imagine the farm, the truck comes in, do the treatment, and then move on to the next farm. So just go around essentially. So the technology now has been incorporated, accepted into New Zealand's greenhouse gas emissions inventory reporting system. This is the first year it's gone into. And Alistair tells me it's also into overseer. Thank you, Alistair, for your team's hard work in getting into the overseer system. It means the farmer who is using the technology, they can claim a carbon credit, or whoever is funding the system, the application, they can claim a carbon credit. So if every dairy farm uses it, you can reduce about 1.2 million tons of CO2 equivalent. So that's about similar to taking off 250,000 cars off the road. I mean, petrol cars, not electrical cars. Okay, so it's a substantial reduction. I think it would be great if the technology is actually incorporated into the emissions trading system. At the moment, agriculture is not in it. I think that they should accept technologies that is available, viable into the emissions trading system. But I'm only a professor, so what I think doesn't mean nothing. We've also measured what happens to the phosphate when the effluent is spread onto the farm, whether it's treated or untreated, simulating some tile drain systems or larsimeters, both shallow leaching and deep leaching. And you can see if you apply the effluent untreated, there's quite a high concentration of dissolved reactive phosphate. So these are solubilized phosphate draining out of the soil into the waterways. You treat it with the ingredients, there's hardly anything coming out. More than 90% reduction in phosphate addition losses because the iron added has reacted with the phosphate, forming iron phosphate, which is a solid material, so not solubilized. And similarly with the total P, also over 90% reduction in phosphate addition losses. E. coli, so the acid and the iron, they interfere with the bacteria, E. coli. They essentially rupture the membrane of the cell, so they actually kill them as well. So you can see the numbers are reduced by 99.9% in the E. coli numbers. So I often see after the treatment, the water, the effluent almost drinkable. We haven't tried it. It's getting very safe. So you get 3 benefits. You get a methane reduction, you get a phosphate reduction, and you got an E. coli reduction as well. So it certainly will protect your surface water from your effluent sources. So just in summary, we quantified the sources for nitrous oxide and nitrate leaching. Urine patches are the dominant sources, and effluent can be a major contributor for phosphate and E. coli leaching. We looked into the ammonia oxidizers and found that bacteria is the dominant community responsible for ammonia oxidation and the urine patches. Archaea doesn't do much. And then common mox is also there, and they play a part, but they can all be inhibited by nitrification inhibitors. So by inhibiting these microbial communities, ammonia oxidizers, we can significantly reduce nitrous oxide emissions and nitrate leaching. And by treating your farm dairy effluent, you get all the three benefits: methane reductions, phosphate leaching, and E. coli leaching. So essentially, we have kind of taken care of a large part of the environmental problem from the grazed pasture systems: nitrate leaching, nitrous oxide emissions, phosphate leaching, E. coli leaching, and methane emissions from the effluent pond. That is all from me, and thank you for listening. I'm Keith will be happy to answer any questions.
Thank you, Lee, for such a wonderful talk, and it's fun.
Thank you.
I open up to the audience to ask questions now.
Lee, thank you very much. Very interesting talk. Really good to see lab fundamental science actually making its way through to impact on that. So, Keith, I see with the nitrification inhibitors, I see a strong parallel here with target site resistance in herbicides. So target site resistance is where we have a herbicide is targeting a single enzyme in the plant and blocking that. And I'm seeing exactly the same with these nitrification inhibitors. So my concern here is if we're using the same nitrification inhibitor, the same mode of action as we say in herbicide resistance, what is the potential for the bacteria, particularly here, to actually start developing resistance to these nitrification inhibitors?
That's a very good point. Actually, we looked into this aspect into DCD as well. I think the difference between the nitrification inhibitors, and we are trying to find one that works in this way. So we're not trying to find a bacteria site. It doesn't kill the bacteria. We're trying to find an enzyme, a compound that only blocks the active site, so that essentially the microbes lay dormant. So with the DCD, we did not find much resistance. And the current one that we've just developed is, we call it a suicidal sort of binding mechanism. We will certainly look into it. Hopefully, it wouldn't happen. I think if it's like an antibiotic or bactericide, it's probably more likely to develop the resistance more quickly. Whereas if it only targets the ammonia oxidizing enzyme by blocking the active site, it may not develop so quickly. That's our understanding. Very good question.
Every year, at least several dozen sewer workers in India die from phosphate poisoning, and that's the other sink for protons besides methane, ammonia, and hydrogen sulfide. Have you looked at the mechanism by which this reduction of phosphorus might be occurring in your eco pond? Because it could be a health and safety issue.
Sorry, you mean the production of hydrogen sulfide?
You show production of hydrogen sulfide, but what you haven't shown is any information you might have gathered about the production of phosphate.
Oh,
H3P, which is commonplace in sewer gas.
Yeah.
And the organisms that produce it aren't selected against the way they are in the room. So I'm just wondering whether that's a possibility.
In hypothesis, we haven't measured it. You have, in theory, because we are reading the rate of potential of the pond, probably the reduction in phosphate is probably even less likely. So the reduction in phosphate happening under more anaerobic conditions, whereas by adding a compound to raise the redox potential, you make less likely to be reduced. So the phosphate now is more preferably reacting with the iron added, forming iron phosphate.
I see. Thank you.
Yes.
Yeah, the iron addition is essential to this process because it's the iron that preferentially reacts with phosphate.
Thank you, Keith.
Good job, mate. Very good.
An interesting question. We heard Jacqueline speak last week about various aspects of nitrification and that sort of thing. And one of the things that she identified, and correct me if I'm wrong, but she indicated that our estimates of nitrogen and urine was a lot longer than what we thought it was. She presented some US data, I think it was, and indicated that that was where we got this 1,000 kilos from, or something like that. Can you just give us an explanation of what your understanding of the amount of?
Yeah, thank you. Good question. Good question. Sorry, I missed Jacqueline's talk. Unfortunately, I wasn't here. So, in any case, we and also others in the CRIs measured the nitrogen concentration and the nitrogen loading rate on the urine patch many times. I only showed you one graph. I mean, we collected, I don't know how many times, the urine from the animal, the dairy cows, to follow them around and measure the volume, measure the nitrogen concentration in them many times. It obviously varies widely. You know, I can tell you from my own experience, sometimes you catch maybe 2 liters, other times you catch 5 liters, other time you might have 10 liters. So the volume varies a lot depending on how much water the animal drank, and also the topography. You know, what is the micro topography on the surface of the soil? You know, if you have a little slope, it might cover like a big area. You know, if you have a little valley, it might cover a slightly smaller area. So the concentration and the area coverage varies a lot. But usually it'll be varying somewhere between, for a dairy cow urine patch, probably between about 400 to 1,000 or thereabout. So when we do experiments, we try to usually use sort of somewhere 600, maybe 700 kilograms per hectare, trying to see somewhere in the, you know, in the middle. When you do research, you sometimes also make sure, you know, you are not underestimating the problem. If you're able to treat it, even if the issue is very serious, that means if the urine patch is even lower, it will work even better. So it'll be somewhere between that. For sheep, it will be much lower, more like 200 or 300. Yeah. But for the dairy cattle, it's certainly much higher. Essentially, the problem arises because to grow the maximum yield for the pasture, white clover pasture, you need to put a lot of nitrogen fertilizer on. And the nitrogen concentration in that mixed pasture is usually about 4% to 4.5%. That's the optimum nitrogen for the pasture to grow to reach the maximum. The dairy cow only requires 2%. Okay, so if you feed, for example, maize silage, your nitrogen concentration will be much lower than if the cows were grazing the grazed part of the ryegrass white clover pastures. So that is the key problem. So that the cow then excretes the surplus nitrogen not required by the animal itself. Yeah.
You have a question online. What effect does application of nitrification inhibitor have on dry matter production for pastures that do not have any urine on them?
Well, the work was done mainly focusing on there are urine patches. I mean, if there was nitrogen in the inter-urine patch areas, we also found it increased the pasture production as well, because there's nitrogen cycling taking place all the time. There's mineralization, immobilization, and you also apply nitrogen fertilizers. Essentially, what happens, the inhibitor is like you have a sink on the farm, you're basically partially blocking it. It doesn't matter where the nitrogen is, if you're blocking the sink, you basically reduce the leaching losses and the gas emissions. So we did find increase the past production, and Jim Moy can tell you that in more detail, even in the inter-unit patch areas. Thank you.
We were naively thinking that the technology would be taken up because of reducing nitrate leaching. But the technology was taken up because the farmers grew more grass. And we got 10, 15% increases, and that was the driver. And the side benefit was reducing nitrate leaching and greenhouse gas emissions. That was the side benefit. It just shows the importance of making sure the technology we develop has a business case and a value proposition for the farmer.
With DCD, we used to do 2 applications in sort of autumn and midwinter. What with the new, more potent compound, what do you envisage might be the application regime?
Thank you, Alison. Great to see you. And we have developed a very novel system to apply the new nitrification inhibitor. I'll tell you all about it in the next talk.
First of all, I want to thank you for the talk. Obviously, I should have brought 2 bottles. Thank you very much. Keith is more than happy to answer some questions as we go out and have nibbles and drinks. So please join us in the foyer. Sorry.